Hello and welcome to this quick guide on how to write a sequence on Excalibur. So first of all, what we want to do is navigate into the lab directory, in this case Canarak, and our name folder. I'm Andrew, so I'm choosing Andrew. And then we do the folder for this project by year, month, and day. And that directory is very important. It's important that we follow this protocol because it's how we organize our entire setup. The base file name starts the same way, year, month, date, and then the machine that the sequence is running on, QEF2 in this case, and then the column. So I'm running on C18. And then finally, we want a kind of descriptive indicator of what our experiment's about, just for ourselves. And the path is going to be, for the result files, the same one we just set up, right? And the instrument method is whatever method in Excalibur methods folder, yep, in this area, which you're going to use for your experiment. The tray type has to match the tray and the autosampler, which you're going to put your samples, and the number of samples box there indicates the autofill row numbers that it will give you. First thing, we're going to change the injection volume. I'm going to use 1. It's a typical injection volume, but it depends on the experiment. The first position at RA1 is always a blank. And then we're going to set up based on the MS database code. So for me, Andrew Colhane, AC. And down the column, we're going to auto-populate, and it will basically start from whatever code number you give it. So these are the samples you prepared. And then we're going to finish with our pools. Now we're going to add a few more blanks in here. We always want to have a few blanks just to let the column do its thing at the start of a run. Equilibrate, get any air bubbles out, etc. Keep things moving. And then we're going to add a few at the end for the same reason. Clear out the needle and the source, etc. So now we're going to save our initial sequence, and we're going to highlight that first part inside any of the cells, copy it, go into our folder, and paste the method and save it. So that's the easy way to just save the base of it. And now we're going to export our sequence into the same folder, and we're going to call it _rand at the end, and you copy and paste the first part again from the cell which we got it from. Just for simplicity's sake, it's easier to do that. So now we're going to open it up in our bootleg version of Office. You may have Excel on your computer, and we're going to enter a new column here, and we're going to use the rand function just for these non-pool samples, so they're randomized for fairness and QC purposes, essentially. And so now we have those randoms. We're going to scan across there, highlight across, and then sort ascending, descending, it does not matter which. Same difference. And now we're going to delete that column, and we're going to save this CSV again. Now we're going to import that CSV. And so we import here, we will use the original positions to put the actual samples obviously in the wells and the autosampler, but we will double check physically that the randomized positions in this sequence line up. Now here you see I'm clicking on 24. I'm going to put the pools again and inject them at the start. I have to click on that little arrow to the left of the 24. Otherwise, it will only copy the first row, not all the rows. It's a weird thing, but you have to click on that. You have to make sure you click on that. And then you can scan down and it will highlight everything in the rows below. So we're just blanket copying and pasting all of those there. But it's very important that you click on that arrow before. And then we'll insert a few more here. And just speed things up again. Yeah, inserting some more to create space there. It will create an additional one of the cell above. And then you paste those in there, double check that all the cells are being filled properly. And we inject pools multiple times for QC purposes, and we'll inject, if we can, P1X and P01X in the middle every 8 to 10 samples. So I'll insert new rows, just copies the ones above, and then you can override them. Again, when you're copy and pasting cells, remember to click that funny arrow to highlight the whole set of rows, so you're pasting the whole thing. Then we'll overwrite there, so now I have 3 sets of P1X and O1X. P1X and O1X are the most important, 1X being the most important to inject multiple times. If you don't have enough liquid for all 3 to be injected, generally, and O3X is the least important. But you want to make sure you have at least 1 of O3X. And we'll inject UCRES UAAs, are standards in each run. They're unlabeled standards, which we just give us an indication of run to run how the machine's doing. The signal from those. Position is important to update. Sample ID is not, but the finicky part of my brain likes to make them match up in both anyway. You can also hide sample ID column because it can be a little confusing and it's irrelevant, but anyway. So we'll save now and run. We're just going to do the same name, _rand, because it's the randomized sequence, the fully matured sequence, if you will. And we're going to run and then click standby after the run, always. And well, that is the brass tacks shotgun guide on how to write a sequence in Xcalibur in the Canorac lab. A quick last thing that I haven't shown here, you will probably want to add a little uniquifying letter or number on each non-unique pool injection, otherwise they'll get this sort of ugly timestamp in the results file, so you know P1XA, P1XB, etc. That's pretty much it. Hope you enjoyed this guide.